Welcome to the Outrun the Sun On-Demand Melanoma Forum. We will hear from our most recent melanoma research scholars. Please watch these brief presentations sharing current research aimed at better understanding skin cancer and developing innovative therapies to treat patients with melanoma. We are pleased to introduce Shuyang Chen, PhD from the Cleveland Clinic, Roxana Perker, MD, the University of California, San Francisco, and Helen Diller Family Comprehensive Cancer Center, and Jeremy Chang, MS, MD, from the University of Iowa Hospitals and Clinics. We will hear first from Dr. Shuyang Chen. Dr. Chen's lab has primary research interests in the environmental and genetic factors contributing to melanoma development. His research is focusing on the reasons why melanoma disproportionately affects fair-skinned people with blonde or red hair. Dr. Chen explains how his research is gaining insight into the mechanisms of melanoma development that will lead to strategies to combat melanoma.
Hello everyone, my name is Shuyang Chen. I am an assistant professor in the Department of Cancer Biology at Cleveland Clinic. My research focuses on melanoma, the deadliest form of skin cancer. Today, I will be giving you a brief introduction to my research on this aggressive disease. To start, what is malignant melanoma? Melanoma is the deadliest form of skin cancer. There are three main types of skin cancer: squamous cell carcinoma, basal cell carcinoma, and malignant melanoma. While squamous and basal cell carcinoma can spread if untreated, they generally grow slowly and are less dangerous. However, melanoma is different. It grows rapidly, is highly aggressive, and can be life-threatening. Why is melanoma so dangerous? Though melanoma accounts for only 1% of skin cancer cases, it causes around 80% of skin cancer deaths. Its progression from being curable to life-threatening can happen in as little as 6 weeks. The reason is that melanoma can easily metastasize or spread to other parts of the body, making early detection very crucial. For example, in the early stages, the 5-year survival rate for melanoma is 99%. However, once it metastasizes to distant organs, the survival rate drops to just 27%. So, what are the risk factors for melanoma? There are several, including family history and skin that burns or freckles easily. One interesting factor is fair skin, especially in individuals with blue eyes, blondie, or red hair. This tells us that pigmentation is a major risk factor for melanoma. Now, let's talk about pigmentation in more detail. One important gene that regulates pigmentation is the melanocortin-1 receptor, or MC1R. This gene plays a key role in determining hair and skin color, as well as melanoma risk. For instance, individuals with a functional MC1R gene tend to have dark hair and skin, which provides some protection against melanoma. However, those with a variant of this gene, often associated with red hair, fair skin, and freckles, are at a significantly higher risk of developing melanoma. My research focuses on how to target MC1R to reduce melanoma risk in people with red hair variant. In previous studies, we identified a small lipid molecule called palmitic acid, which can increase MC1R function. This happened through a process called palmitoylation, a post-translational modification of the MC1R protein. This modification boosts the gene's function, helps to enhance pigmentation, and potentially lower melanoma risk. To better understand this process, we conducted numerous experiments, including creating a genetically modified mouse model. We found that mice with the red hair variant of MC1R had golden hair and a much higher risk of melanoma compared to mice with the wild-type black-haired variant. From this research, we learned that palmitoylation is crucial for proper pigmentation and melanoma prevention. When the MC1R gene is fully palmitoylated, it produces black pigment and promotes efficient DNA repair, offering protection against melanoma. However, in individuals with the red hair variant, palmitoylation levels are lower, leading to red pigment, inefficient DNA repair, and a higher risk of melanoma. Currently, we are exploring ways to enhance MC1R palmitoylation to further reduce melanoma risk. Recently, we identified a protein called ZDHHC13, an upregulator of this process. Our ongoing research is investigating how we can target ZDHHC13 to increase palmitoylation of MC1R and ultimately find a new way to reduce melanoma risk, particularly for people with the red hair variant. Thank you for your time, and I look forward to any questions you may have.
Next, we will hear from Dr. Roxana Perker. Dr. Perker's research project concentrates on understanding how malignant melanomas may arise from benign pre-existing nevi, and in particular how mechanisms associated with cellular senescence may prevent melanomagenesis. The objective of Dr. Perker is to elucidate the signaling pathways that cause senescence bypass and immunoevasion in order to rationally design new treatment strategies and effective therapeutic interventions.
Dear Outrun the Sun team, supporter, and friends, my name is Roxana Perker. I am the 2024 Outrun the Sun Research Scholar. As a dermatology resident from Vienna, Austria, I joined the Helen Diller Family Comprehensive Cancer Center at the University of California in San Francisco last year. The research I lead in the lab of Dr. Coupe focuses on exploring the role of cellular senescence in preventing melanomagenesis. I work on identifying how the tumor suppressive mechanism of cellular senescence can be corrupted and enable a benign mole to transform into a nevus-associated melanoma. By understanding this, our objective is to contribute to precision medicine in three main ways: improve cancer prevention, provide a more refined diagnostic classification of melanomas, and design more effective therapeutic strategies to treat this group of melanoma patients. As a reminder, cellular senescence is best known as a cell-intrinsic mechanism that prevents cancer by limiting the proliferation of damaged or malignant cells. You can imagine that this protective, tumor-suppressive effect of cellular senescence is critical to induce and maintain the irreversible growth arrest of melanocytes that experience an initial cancer-like surge in cell division. This is triggered by oncogenic mutations such as NRAS or BRAF, which subsequently lead to the formation of a nevus. If the senescence process didn't happen so systematically, then every single oncogenic hit would transform a melanocyte into a cancer cell. Despite this highly protective and effective tumor protective mechanism, the problem is that even though exceptionally rare, nevus-associated melanomas still account for around 30% of all melanoma cases. 60% of melanomas are so-called de novo melanomas, which arise in previously unremarkable skin, and a very small group that develops in tissues that are probably representing a very different kind of melanoma altogether, such as mucosal or uveal melanomas. How to transition from a nevus to a nevus-associated melanoma happens still remains unclear. The underlying idea for our project is to utilize such unique clinical conditions in these very early steps of melanomagenesis as a mean to better understand the mechanism of senescence bypass and immunoevasion that might drive cancer initiation and then progression. To address this, we're leveraging a spatial transcriptomics method to define gene expression profiles at the single cell level in patient biospecimens. Our goal with this is to characterize differences between nevus and its adjacent melanoma. We're particularly interested in pathways that may help normal melanocytes bypass or escape cell cycle arrest, and how this may start promoting their growth and survival. To further understand the process, we're also using a high-throughput kinase activity mapping technology, which has been developed in this lab right here. It will help us elucidate whether potential defective or hyperactivated kinases and their phosphosignaling circuits cause melanocytes to fail to senesce and instead bypass the otherwise irreversible tumor-suppressive senescence arrest. To compare kinome profiles, we're using paired nevus-associated melanoma biospecimens, as well as a primary cell culture model that we induced to senesce or bypass senescence arrest. All this goes to show that nevus-associated melanomas are uniquely and most importantly accessible clinical case to identify the essential mechanism that underlie the nevus to nevus-associated melanoma progression. Our subsequent goal is to translate these transcriptomic and kinome analysis into the most effective and clinically promising drug combination strategies. We plan on ranking so-called top hits to test synthetic lethality by cell survival-based drug screening. We're especially interested in BRAF and NRAS activated cell growth dependencies that may involve CDKs and potentially the PRB pathway. Co-inhibition of the most promising targets should ultimately either induce cell death or even restore senescence growth arrest in these nevus-associated melanoma cells. With tremendous advances in skin cancer treatments across the board already, melanoma therapies still remain one of our biggest challenges to date. Your support is helping me tremendously in working on this research project and hopefully being able to share it with the rest of you all very soon. For now, I would like to thank you not just for your support, but also for all the work that the Outrun the Suns are doing. I think it is wonderful to be brought together by such an important cause like keeping our skin happy and healthy. I hope to meet you all soon at one of the Outrun the Sun events, and in the meantime, bye from me.
Next, we will hear from Dr. Jeremy Chang, a general surgery resident planning to pursue a surgical oncology fellowship. His research interests include investigating the role of cellular plasticity and heterogeneity in melanoma metastasis formation. Under the guidance of his mentor, Dr. Colin Kenny, he is working to define the significance of the MITF-TFE transcriptional axis in invasive melanoma. He is additionally interested in using machine learning algorithms and radiomics in improving diagnostic and clinical outcomes and medical education.
Hello, my name is Dr. Jeremy Chang. I am a fifth-year general surgery resident at the University of Iowa Hospitals and Clinics. I work as a postdoctoral researcher in the labs of Dr. Colin Kenny and Dr. Ronald Weigel. I completed my undergraduate training at the University of Pennsylvania, receiving a triple major in biochemistry, biology, and economics, as well as a master's of science in chemistry, completing my thesis work in the lab of Dr. Mark Kahn, studying the transcriptional regulators of lymphatic development. I received my MD degree from the University of Tennessee Health Science Center before coming to Iowa for my general surgery residency. Our lab's research interest is investigating transcriptional mechanisms governing melanoma cell fates as it relates to intratumoral plasticity. Melanoma is the most lethal cutaneous malignancy, while the 5-year overall survival of localized and regional melanoma is 98 and 63% respectively. The 5-year overall survival of metastatic melanoma remains a dismal 5 to 20%. This is despite improvements in chemotherapy and immunotherapy. An important characteristic of melanoma is its cellular plasticity. Melanoma has the propensity to convert from a proliferative, pigmented, and relatively immotile state to a migratory and invasive mesenchymal state. This transition has been referred to as phenotype switching in the field of melanoma. The design of novel therapies for metastatic melanoma depends on our understanding of the mechanisms that govern this transition. Microphthalmia-associated transcription factor, or MITF, plays a role in physiologic development and also differentiation and survival of melanocytes. In malignant melanoma, studies have shown that high levels of MITF activity are correlated with a proliferative and melanocytic phenotype, while low levels of MITF activity are associated with increased metastasis and drug resistance. The mechanisms governing the invasive genetic signatures in MITF low melanoma, however, are poorly understood. To investigate the mechanism of upregulated gene expression in MITF low melanoma, we performed cut-and-run sequencing and RNA-seq in wild-type SKML28 and SKML28 MITF knockout melanoma cells, and this was to investigate how and where MITF interacts with the DNA. We found that in MITF knockout cells, there were in fact thousands of novel anti-MITF antibody binding sites, which were absent in the wild-type cells. Utilizing our gene expression data from RNA-seq, we found that these novel binding peaks were associated strongly with upregulation of genes involved in cell migration and EMT transition. Our further studies showed that the anti-MITF antibody utilized in the cut-and-run experiment also bound to binding sites of other transcription factors in the same basic helix-loop-helix zip transcription factor family. Through computational analysis, we found that the transcription factor most significantly associated with binding at these novel peaks was in fact MITF paralog transcription factor E3, or TFE3 for short. We further confirmed that TFE3 was the major transcription factor associated with this specific invasive pattern of gene regulation by creating double MITF and TFE3 knockout cells in the SKML28 cell line and performing anti-MITF anti-TFE3 cut and run and RNA sequencing, which resulted in loss of these previously described peaks and gene expression changes associated with downregulation of an invasive gene signature. This was additionally confirmed with similar experiments in the A375 melanoma cell line. To investigate the role of TFE3 on melanoma phenotypes, we knocked out TFE3 in characteristically invasive MITF low A375 melanoma cell line. We performed in vitro transwell invasion assays as well as in vitro wound healing assays, which demonstrated significantly decreased cellular migration with TFE3 knockout. We subsequently performed in vivo injections of cells into the lateral tail vein of mice and found that TFE3 knockout cells lack the ability to colonize and form tumors in the lungs of immunocompromised mice, further demonstrating loss of metastatic capacity. We've been able to generalize these phenotypic phenomena by studying a panel of human melanoma patient-derived xenografts, knocking out TFE3 and demonstrating the same loss of invasive potential occurring in these cells. So as a summary so far, we've been able to show that MITF low invasive cells, MITF paralog TFE3 promotes expression of an invasive mesenchymal-like transcriptional gene program. The next question to answer is what the mechanism by which loss or downregulation of MITF in fact upregulates TFE3 activity. We suspect that there is some antagonistic interaction as there is an inverse relationship between MITF and TFE3 protein expression in melanoma cell lines. Previous studies have actually shown that in naive pluripotent stem cells, a complex containing folliculin and folliculin interacting proteins 1 and 2, FNIP1 and 2, promotes cytoplasmic retention of TFE3. Uniquely in our data, we found that FNIP2 is directly regulated by MITF. This pathway functions through regulation of mTOR-mediated phosphorylation of TFE3, which subsequently exposes a degron for culin ring E3 ubiquitin ligase 1, or CUL1, which targets TFE3 for lysosomal degradation. We validated this pathway by treating cells with taurin-1, which is an mTOR inhibitor, subsequently showing that inhibition of mTOR leads to increased nuclear localization, indicating increased TFE3 activity. These findings overall demonstrate a novel mechanism of transcriptional factor regulation that plays an important role in melanoma tumor aggressiveness. Modulation of this pathway may prove insightful in developing new treatments for metastatic melanoma.
Thank you for watching the presentations from the most recent Outrun the Sun melanoma research scholars. We are honored to support 33 melanoma research scholars working to develop innovative therapies to cure melanoma. Funding also supports educational programs that help prevent skin cancer altogether. Outrun the Sun is dedicated to building national awareness of melanoma and other skin cancers, educating communities about preventive measures that reduce melanoma's incidence rate, and to raising funds for melanoma research leading to effective treatments and a cure. Learn more at outrunthesun.org.